Hi. All right, we're going to talk about tools.
Don't sound so excited.
Oh, sorry. Kick it back a little bit. Okay, so these are sort of, this session is really trying to address some of the looking forward recommendations from the 2014 report. In particular, to read this, developing a strategic plan for continuous updating of the IRIS methodology. Strategic plan should address applying advances in data retrieval and text mining. Committee also found proposed format assessments should enhance our user friendliness and transparency, and references evidence tables and data displays. And so what we're going to show you now are sort of our advances in using specialized software applications to help conduct systematic review. A few ground rules. So the specialized software tools make the process more efficient. There's clearly sort of time and cost savings, improved data management, increased transparency. It is incredibly important that you recognize that when we talk about software tools, we are not saying that it's push the button, go, and you get a result at the end. Some of the tools that Michelle's going to be talking about are really designed to sort of help facilitate the process of evidence identification. I'm going to be showing you an application called Hawk that has sort of a, there's nothing automated about it. We would love it if it was automated. It's a software application that just sort of helps structure the data extraction when you summarize study methods and results. We think it sort of gives you some really nice visual capabilities to make it easier for assessment teams and reviewers to look across study pattern and visualize findings. Not automated. But we are, and I'll talk about this a little bit more, we are investing in sort of a research effort along with NTP, along with others at the EPA, to continue to sort of look for ways that we can keep automating or semi-automating the process, always sort of anchored at assessing the performance. So within the IRIS program, we're going to be highlighting some of the software applications that we use today now. The ones that Michelle is going to be talking about, Swift Review, is free to everybody. Another one, Swift Active, is sort of still a bit beta-ish, but free to feds. Hawk is free and open source. And so there's a theme here. We are trying to find tools that are free when we can, because we really see this as sort of not just something for our teams, but we really think that this is a service that we can provide to the field, and not just the environmental health field, but also sort of the biomedical field in general. So we're deliberately trying to sort of keep an open mind to identifying tools that fit within our workflow. When there are options available and there are free tools that work well and they can integrate with Hero, we prefer those. And then I would say that, and we've mentioned this a little bit earlier, in terms of how we sort of got from January 2017 to the point where we're at today, where we actually have quite a few people within the IRIS program who are feeling very comfortable using these tools. Was we just started off with some sort of deep dive training retreats, coming up with instructional videos, identifying a group of people. I forgot to introduce you guys. Michelle Taylor, Amina Wilkins, Xavier, you've met. And then we have others who are in RTP, not physically here today. A team of people who become really conversant in these tools. So that when an assessment team is at the point of saying, hey, I'm screening, I need help, I need to use Hawk, I need help, I know I took that training, but I can't remember what you said, can you help me? Then we have sort of this train the trainer model so that we can provide to the support to the teams when they need it. In terms of the research activities, so most of the mature tools that people feel, and not just us, people in the field tend to feel comfortable using now are really, again, around evidence identification, mostly sort of bag of word models to help priority rank studies for screening. A longer-term goal that we and others have is to help automate or semi-automate beyond that. It's a very long-term research commitment. A major hurdle is a lack of training and test sets for model development. So completely unsexy work, but what we're doing now in partnership with Andy Rooney, NTP, and others at EPA are trying to go through the tedious process of developing those test sets, which involves having a PDF or something like that and annotating it so that you can say, species, this is the content in this paper, and then you can give that to model developers so they know what to aim for. So that's where we're at now. We think with sort of the data extraction side of it, which is sort of this next frontier, It's going to be hard work. We think it's going to be a little bit easier for more experimental designs with more of a structured format. So we're focusing first on the animal bioassay, the epidemiology studies we think would be even trickier. We think any progress that we can make would be helpful, even if you can sort of pre-populate, and this will make more sense after you see Hawk, pre-populate fields like species or test chemical or purity, and have a person check it. That's still, on balance, when you think about the number of studies that we do, it translates to a time and cost saving. And just for perspective, to do data extraction on a single animal or a single epi studies, you're looking at about 1 to 3 hours. And you're typically looking at people who have a lot of experience, especially when you're talking about evaluating the quality of studies. So huge time and cost sink. So to help advance this element of our research, in 2017, we created, I know nobody will believe this, a funds out interagency agreement with NTP, where when we can find pockets of money in EPA, then we can work through NTP's sort of more considerable resources and help advance this sort of dual mission. Again, our current work is really focusing on this process of trying to develop these test and training sets. Other parts of EPA can also utilize this interagency agreement. So Christian Markey from OCSPP, Office of Chemical Safety and Pollution Prevention, is also heavily involved, and that's where Tosca lives. So he's heavily involved in thinking about how do we advance this automation portfolio. Dale Hoff, who's director of the Mid-Continental Ecological Division, he manages something called the Ecotox database, which involves curation of ecotox information. So another keen, interested partner in tools to help identify the evidence and summarize it. So we have sort of, we have some real strong connections within the agency, and then I think this partnership with NTP. So we expect this to be sort of, at some point, a very fruitful engagement, but it's going to be, when we get to the data extraction side, it's not going to be a 6-month deliverable. We think that some of this work, some of the more easier to find content, can you tell me what the test chemical was, the species, the sex? We think that this sort of developing the training and test sets and then the model development might be facilitated by FTEs and some contract arrangements, but at some point we envision innovation challenges, especially when you're trying to find content that spans paragraphs or is in tables. That's the really hard stuff. And I think, actually, you.
So we are currently using a suite of available systematic review tools, some of which are going to be talked about tonight and demoed tonight. I'm right now going to talk about, and these tools are all interoperable. I'm going to be talking about Swift Review, which is a publicly available free desktop application. So you download it to your computer. It works with both PC and Mac. And it's used during problem solving and scoping. Users are able to bring in data from either Hero or PubMed, and they're also able to bring in output from your reference management tools such as EndNote. And it uses a 4-pronged approach where search refinement discovers important terms and phrases. It uses an open source software, Apache Lucene, so that's freely available to anyone. And it provides search engine and query search strategies using Boolean and wildcard operators and fuzzy searches. Another prong is topic modeling, which helps uncover unhidden structure within your literature corpus. Also literature prioritization, which is the machine learning component of Swift, and also problem formulation, which helps identify your research questions. So you can use you can screen studies according to your PICO. And it has built-in and user-defined search queries that allow you to target your surveys. And it uses machine learning, as I said before, to prioritize literature, reducing your screening burdened by at least 50%, sometimes more. So Swift review tags are labels that are annotated to your bibliographic documents. And then they are then organized into tag categories. For example, a tag category for health outcome includes the following tags such as cancer, cardiovascular, or neurological. When used with your tag browser, which I'll show you, or your search functionalities within Swift, these tags facilitate increased efficiency during scoping and problem formulation by quickly finding the documents you're interested in, and these tags can be assigned both manually or automatically. So the user has control. So these tags can be assigned either by mesh terms or your bibliographic data. So for example, if you're bringing in from PubMed, along with that comes the associated Medline tags, including your mesh terms, publication type, pharmacological actions. You also have queries and filters that incorporate several of the defined search filters. These filters have been prepared by information scientists for various topics of relevance to environmental health scientists. And details on these filters are documented, so you can, very transparent, just go to help and go to search strategies and you can find more detail on that. You can also tag by chemical name, automatically tagging documents that mention chemicals occurring in several chemical lists of relevance to environmental health researchers, such as the Tox 21 list of the 10,000 chemicals. So here's just an example of some of the built-in user strategies. This is for cancer. This is from the report of carcinogens. And this is several pages long. This is just a screenshot of the first page. You can also search within the tag browser, and you can, within that browser, define tags that aren't already built in, so you can customize it. You can also, here's a list of some of the example search terms. You can find those within Swift by going to the Help tab next to the query. You can also bring in established search strategies that you've already developed by doing a batch query. So this is just an example of a screenshot within Swift. To orient you, the top left panel is your navigation pane that includes your tag browser, your search query, and your batch query, your mesh term tree, and your prioritized list. On the top right is your data pane. You have your document lists, the pie charts and the bar charts. And on the bottom is your document list. That's where you do your machine learning. So this is just another example of that previous slide showing the same information depicted as a pie graph. So this is all using a graphical user interface. By selecting any component of this screen, so if I go to the navigation pane to the top left panel and select cancer, I bring up those documents that have been tagged cancer, and it will auto-populate that bottom document panel. If I go to the pie chart and select any of the pie slices, I can do the same. I can bring up additional levels. So here I've brought in the tag categories for the Tox 21 chemicals. You can also bring in another level by evidence stream, so by animal, human, and in vitro studies, it'll tag them accordingly. You can also highlight keywords to facilitate your screening. So you can manually enter in the keywords that you want highlighted within the abstract and the title to help facilitate the screening. It also produces interactive displays to reveal patterns of evidence. This is a heat map browser. And on the rows on the top left navigation pane, I have animal, human, and in vitro as my rows. And my columns are the health outcomes. You can change that to display health outcomes on the rows and maybe the Tox 21 chemicals for your columns. And again, I can select any of those boxes and bring up those relevant studies in my document list. So this is an example of health outcome tagged heat map. And I've binned these by 5-year intervals. You can adjust that by however many years you want to bin them. If you do like an update to an assessment and you want to look at the most recent publications since that assessment, you can do that and reveal those documents that have been published since the last assessment. So you also can do priority ranking. This is the machine learning component of Swift. You can have topic modeling. That's a statistical method of automatically computing the categories for your documents. Users can also build their own topic model. Can incorporate human-curated training sets. So you can bring in a training set of documents that you know are going to be included or excluded. Or you can go ahead and manually tag as you go if you don't know. So this is an example of how you would go about training your model. So there are 2 columns there, not sure if you can see them. The second from the left in the document panel on the bottom is your training item. And then the column next to that is whether or not you're going to include it. So if I select the training box and then also the included box, that's going to prioritize all those documents that are similar to that, move them to the top so they're screened sooner. If I select training item but don't select the included box, that'll be one of the documents that'll be excluded. Similar documents will then be pushed down further. And this is just a graphic depicting the performance of the priority ranking and how it improves the efficiency of your screening. So the yellow bar line is the performance if the screener was going to randomly screen. And the blue line is the performance using priority ranking. We have 100 documents there. By randomly screening, we reach 100% inclusion of 100% of our studies. And the blue line, you can see after screening 10 studies, you've already reached that 100% inclusion of 100% of your studies. So that dramatically decreases the amount of time you're spending screening. So results from that can be pulled directly into Swift Active Screener, while Swift Review doesn't help you screen because you still have to manually annotate your training seeds. Swift Active Screener does not require those training seeds, and it helps you screen. It's a web-based, real-time, collaborative, systematic review software application. It's using state-of-the-art statistical models to prioritize articles as they are being reviewed. I'll show a couple screenshots of what that looks like. Experience suggests that screening burden is reduced by at least 50%, likely more. And the algorithm improves from screener input without the training seeds, as I mentioned, further increasing efficiency, more efficient than implementing a seed study-only model. The option to seed exists, so you can, if you know you have an article that you know is going to be included, you can go ahead and seed that, and that'll just improve the efficiency of the model. It incorporates, again, like Hawk, like Swift Review, that graphical user interface, so a lot of this stuff is point and click. You can hover over and select all over the visual. In its user-defined screening levels, you have a level 1, title and abstract, level 2, full screening, and then you can have a conflict resolution component. So here is a screenshot of how you're able to customize your inclusion and exclusion criteria or your questions according to the PICO statement. You can invite reviewers here. Typically, we have 2 reviewers, as mentioned. There's a capability here within Swift Active to manage conflict resolution. And here is where well, it's actually the next screen. User input improves the algorithm to priority rank. So it's hard to see here, but you have an include and exclude. If you can see the title. If you want to visualize the abstract, you just hover over the microscope or the magnifying glass, and that will bring up the abstract. This too has keyword highlighting capability, so you could have it display keywords to help facilitate the screening. And this is the option to seed. So if you know you have an article that is going to be included, you just go ahead and select seed, and that will move everything up automatically. You do nothing else. It'll move relevant documents up to the top. So each time you hit seed, it'll automatically revise the document list. You can manage the references with conflict resolution and track and archive changes. So this is just showing you the page where you have conflicts. You can bring it up and see who the reviewers were, what their conclusions were, and you can track this by study, by reviewer, and across the assessment. It also generates visuals that track how long it takes to conduct each of the screening. By seconds, by minutes, it'll display these graphs. You can export these into Excel, into PowerPoint, into your assessment. We use these as part of our project management activities, just to keep track of how long things are taking, who's doing what. So Swift Active is also interoperable, which seems to be the hot phrase today. It accepts imports from bibliographic databases and reference curation platforms, including Swift Review, of course, EndNote, PubMed, and the results from the screening in Active Screener can then be output to standard data formats compatible with the application, including Hawk and Excel and Note. And there are some other users who have been involved during this beta development of Swift Active, NIH, other programs within EPA, TEDx, USDA. And I will be demoing all of this tonight between 5:30 and 7. I hope to see you. Bring all your questions.
Okay, and now we'll move on to Hawk. Okay, so Hawk is free and open source. It was developed at UNC by Andy Shapiro and Yvonne Rusin. Andy Shapiro is now at NIH. They're very, very, very lucky. He's such a fabulous find. Hawk really helps you organize the whole process of systematic review if you want. So it has capabilities to do literature searching and screening. We tend not to use it for that because it doesn't currently let you have more than one reviewer and do conflict resolution, but it has that capability if you don't need that feature. What we use it a lot for is this structured summarization of methods and results from animal bioassay epidemiological studies and in vitro studies if you want. So it has a structured approach, and then what I'll show you is sort of these interactive visuals. It's also, as was indicated earlier by Beth, I think she showed you how we use it to help facilitate the process of risk of bias analysis. And it's modular to work with other tools. So the idea is that if there's only a piece that you want, then that's all you use it for. It works best in Google Chrome. It's a preferred browser, Mozilla Foxfire and Safari. So if you try any of the graphics and you get something broken because it's automatically opening up into Explorer, then you've been warned. So here's just, I can't read this from here, but so here's just sort of what the user sees. This is the data extractor sees. This is what I mean by the structured summarization. What you're seeing here, even though you can't read it, or maybe you can read it and I can't, is information about like chemical purity, what was the vehicle used. This is sort of to summarize an animal bioassay. And then you have other modules that would be, in this case, describing the endpoint, and you have everything from sort of the system level to the organ, to the effect subtype. when you start to sort of type in things here, it's a little bit like Excel where it'll autofill, give you options for words. So it's not a hard drop-down entry for endpoints. It's more of this prompting approach. But we're not going to be talking about today for time, but as we roll this out and think about interoperability with other databases and sort of terminologies or ontologies, we are very interested in trying to sort of promote consistent use of terminologies or synonym finding based queries. So here's an example from chloroform, an epidemiology study. This is just one single study that reported many sort of birth defect outcomes. So this might be a visual, and you can see features of the population, the design, sort of a bit of the exposure. You can put comments in here. It has features of the exposure contrast. And then the idea is that, because I want to find out more, I think this is the encephaly, I want to find out more about how was this particular outcome measured. I can click on that, and then I get this drill down where I see more information about sort of the confounding factors. And so you can do this for each level. You can do this for sort of the exposure assessment piece. How was this done? Again, it takes about 1 to 3 hours to sort of do this process and do the bias assessment. There's a lot of granularity in here. Here's an example for visualizing animal evidence. This is a chloroform fetal survival study, and you're looking at endpoints that might be sort of live litters or early post-implantation loss. And so you're looking at multiple studies here. So this helps you look across studies. And I know you can't read all the details, but it's the same concept. If I want to find out more about how sort of this particular endpoint was looked at, I can click on it, and then I get more details about the methodology. You get also things like, if the information is digitized, collected from a graph, and we had to use a digital ruler to get it, then that would be documented here. There's this column here about confidence. You heard about our approach for looking at study quality, so you could click on that, and that would bring up the sort of the features about the analysis, in this case, a risk of bias for the animal bioassay and the rationales. Animal data can also be expressed as sort of an effect size, so percent control, so we don't have to sort of go toward is the dose level no or low? We can sort of begin to sort of do more effect size. And here's sort of this, in this case, what you're looking at is continuous data, and you're looking at sort of percent control on the y-axis, and then you're looking at dose level. And so this is not from an iris assessment. This is a publicly available example in HAWK, nitrofen. But what you're seeing is individual dose response displays for individual outcome findings. And then this green shading in the back is sort of a relevant exposure range. So you can begin to layer where your experimental data is and have this sort of visualization in the back about how relevant it might be. But the idea here is that if I wanted to find out more about, what's this sort of apparently sort of low dose effect, then I can click on it. And then at the bottom, you sort of see different tags. The study, in this case, is a developmental finding. It's a rat finding. It was found in rats. And there's other descriptors that I can't read from here. But I click on the line, and then you get the same thing. You can find out more about the endpoint, and you can sort of drill down. So the idea is you enter the information once, and then you have all these visualizations to help you present it in the way that's most tailored toward what it needs to be and lets the reviewer sort of drill down. And then you can download the results. So you can download sort of the risk of bias analysis. You can download the results of describing the methods and the results at the level that you could do a meta-analysis on. This is very granular. So this is the idea, it helps with the transparency. So again, people can sort of take this kind of download and do their own independent analyses on it if they want. There is sort of a module in HAWK to do benchmark dose modeling. So we're actively trying to sort of explore this and share this module with our experts in benchmark dose modeling and sort of see how it works in our hands. So I think the advantage is it's, even though it's not automated, a structured extraction process, it'll improve consistency in how we collect information and completeness. It's free and open source and customizable, enhances opportunities for database interoperability. It integrates with many of the tools that Michelle, well, actually all the tools that you discussed. It's web-based, so it really promotes the team collaboration. One of the sort of the challenges in the past without using this kind of tool, if you did bias assessment, you would have to wait for one person to be sort of completely done in order to access the file that they did their own. So having this kind of web-based approach, you can do things in parallel. And then the ability to export data files, it promotes the further analysis, but we also think because we're collecting so much information on the risk of bias, that that's one of the areas of sort of method development in the field, is trying to understand real drivers of bias. And some of them have empirical support, and others it's a little bit softer. So we're hoping that by showing this information, then we're also providing test sets for people to do method exploration on. And then it creates possibilities for moving toward more web-based interactive reports. So, last slide of this session. So hopefully this demonstrates how we've tried to take a real bite out of the looking forward recommendations in the 2014 report. And again, there'll be demonstrations tonight from Michelle in Swift Review, Swift Active. I will be manning the Hawk booth. And then HERA staff will be presenting too.
Okay, great. Thanks. Impressive developments. Where was all this 40 years ago? For those of us who had large file cabinets.